Our next speaker is Matthew Patrick from the Department of Dermatology. Matthew is an assistant professor there. He's going to talk about using MGI for the genetics of skin disease.
So I will discuss how MGI can be used to investigate skin diseases and also their probabilities, and describe in particular our project involving polygenic risk scores and genetic epidemiology, highlighting how the findings from MGI can also be combined with other resources as well. So the goal will be to present how MGI can be used in skin disease research and provide some ideas which may be helpful hopefully to other fields as well. So first I'm going to talk about polygenic risk scores. So this is the work largely of two PhD students who I work with. A polygenic risk score idea is it's a weighted sum of different genetic markers, and these measurements to predict the risk of a certain disease are very important to evaluate the efficacy in different settings and different populations. But most of the PRS are applied to Caucasians, and direct application of those PRS to minority populations is often not very effective. Talking particularly about systemic lupus erythematosus, although over 80% of cases involve skin symptoms, only 25% is that symptom presenting case. So also lupus is highly complex and heterogeneous, which makes it difficult to diagnose often from these different symptoms. We therefore worked in collaboration to create the polygenic risk score and apply it through MGI. We trained a approach called lasso sum on around 7,000 cases and associated controls, achieving area under the receiver operator curve of 0.75 and 0.74 on MGI and BioView, which is another paragraph from Vanderbilt. And interestingly, between these two different data sources, we had very good correlation of the effect sizes with a 0.7 correlation, and this is illustrated by the diagram here, which means that along the y-axis we have odds ratio and printout on the x, so that we can see that as the odds ratio increases also for MGI as it is for BioView. Another thing that we can do, because we have these lab test results which are often used to diagnose lupus, and these are available in MGI, so we can compare the effect of PRS with lab test results. And interestingly, in each case, for the anti-nuclear antibody and anti-double-strand DNA test, including the PRS, improves the performance of prediction even when using the lab test. So another project we did was to apply the PRS to minority populations. There are techniques called Bregman divergences, which can measure the difference between probability distributions, but they often rely on individual level data and require specific probability distributions. So we applied a special case of this approach, which is appropriate for summary statistics. I just highlight that the beta tilde is the effect size from the majority population, and R is a marginal correlation for the minority population. Sigma tilde is the sparse covariance matrix estimated from the minority reference data, and most important criteria, I think, is a ETA, which it tunes between the minority and the majority statistics, so that we can weight it to find an optimal for the target population. And on the simulated data we have, we randomly sampled 20 causal SNPs and set the variance based on the heritability in our simulated data. And we compared our approach, which we called BRIGHT, with other approaches. PRS-CSX is a well-known approach for the purpose of trans-ethnic PRS, and also a single ethnic prediction model, which is called LASSO-SUM, which is either based on the European or African data. And interestingly, across the different heritability setup, we had much better discrimination power for BRIGHT compared with the other approaches. Then applying this to MGI, so we trained the model using summary statistics from the Million Veteran Program, and in these plots the square represents the African American population, and the triangle the European population. So you can see on the x-axis we can tweak the ETA parameter to reduce the error in the prediction, and measured using R squared or MSPSE. So we can find for this there's a particular trade-off in the ETA which will help to reduce the error and perform better than using the African American or European summary statistics alone. And now we're going to give an introduction to some projects we worked on which combine epidemiological analysis with genetics and genomics. First, looking at type 2 diabetes. Importantly for psoriasis, the comorbidities actually it costs more than half of the direct healthcare costs for the disease. So comorbidities is very important to research for psoriasis. And type 2 diabetes was one of the first identified, I believe, to be a very important relationship with psoriasis. Relatively recent research found that the higher the severity of psoriasis, the greater the risk of diabetes. This is epidemiology knows a dose response, so that ideas is a more causal relationship, because if you change one of them, it changes the other one, so that it implies that maybe there's some causal relationship between the two. So we took the summary statistics for diabetes and psoriasis and performed trans-disease meta-analysis between them. For psoriasis we had around 11,000 cases and diabetes 74,000. So due to the imbalance in the sample size, so we have a lot more samples for type 2 diabetes, I took a balanced approach rather than a standard inverse variance weighted meta-analysis. I treated the effect sizes being equally weighted between the two. And now to illustrate the approach using something that isn't a shared locus but looks like it might be. This CDKAL locus is significant in psoriasis. We have here a typical regional association plot with the genetic position on the x-axis and the p-value on the y-axis. So each of those dots represents a specific marker, and the colours show the correlation with nearby markers, so the linkage to equilibrium. When we look at the type 2 diabetes, we also see a significant association in this region, but if we focus on the marker from psoriasis, we see that particular marker and those which are correlated with are not significant. So this suggests maybe two different signals in the same locus. Applying the trans-disease meta-analysis, the type 2 diabetes locus is rejected as it's not significant as in type 2 diabetes, and the psoriasis locus, although more significant in trans-disease meta-analysis than psoriasis, it is rejected because it's in the shadow of the other signal. It is less significant than the other signal, so that means we would reject this particular one as being a shared locus. So applying this approach, we identified 4 different loci which are shared between psoriasis and type 2 diabetes, and then we applied these to the data in MGI, where we had 127 patients who had both psoriasis and type 2 diabetes. And interestingly, we found that the risk allele frequency were consistently higher for patients with both of the diseases than for the controls, suggesting we can, to some extent, validate in MGI data. The overall picture seems to be involving NF-kappa B signaling, with missense variant for choke and EQTL for STAT3, also EQTL for spread 2 and PRDX5. The final project I want to talk about is some research we did for COVID-19. We found that previous researchers suggest there may be some relationship between these two diseases, so we wanted to investigate a bit more. We used data from Michigan Medicine for 400,000 patients. And interestingly, we found that skin diseases, particularly inflammatory skin diseases, had a greater risk of COVID-19, even when adjusted for age, sex, race, BMI, and socioeconomic disadvantage. However, when we looked at the risk of mechanical ventilation, these individuals had considerably less risk of mechanical ventilation, suggesting that inflammatory skin disease imposes a risk of COVID, but maybe less severe COVID. Then we looked at the gene expression in COVID-19 infected lung epithelial cells. We found a reasonably large number of them as being significantly differentially expressed in chromosome 1, particularly in the region for the epidermal differentiation complex. And these are important for psoriasis, and particularly maybe the relationship between psoriasis and infections. So our study found inflammatory skin diseases may increase the incidence but decrease the severity of COVID. The early interferon response or decreased viral load could result in a mild form, and if skin acts as a reservoir, this could result in a different rate of progression compared with direct respiratory transmission. And oral and respiratory epithelium may be similarly affected as skin, facilitating viral entry while preparing the immune system for response. So in summary, MGI provides a useful resource for testing and applying polygenic risk scores. It is coupled with detailed health records that are used in genetic epidemiology. Results can be complemented with other data sources, including BioView or GWAS and expression data. MGI is a key resource as part of a multi-omic approach to skin disease research. I'm extremely grateful for everyone I work with, both from US Michigan and Penn State, and appreciate the psoriasis cohorts and all the patients involved in this research, as well as the funding agencies who supported our work. I'd like to particularly say that I appreciate support from the Dermatology Foundation for my work. Anybody have any questions? Also, feel free to email me your questions for later.
Just sort of completing the circle, do you see a correlation between psoriasis and COVID infection?
Sorry?
Psoriasis and COVID infection? Because I know that psoriasis is correlated with asthmatic conditions, so it affects the lung tissue.
Yes.
So do you think psoriasis and COVID-19, there may be a connection there? That's sort of the third part of your triangle there?
Yes, so that's what we found, but we didn't really necessarily have enough power for psoriasis alone. That's why we focus on for inflammatory skin diseases.
Okay.
We believe psoriasis, but certainly inflammatory skin disease overall.
Overall, thank you.
So thanks for the interesting talk. So this relationship between psoriasis and type 2 diabetes has been around for years.
Yeah.
And so I wonder if you attempted a Mendelian randomization. Weight loss improves psoriasis. Improving diabetes improves psoriasis. Improving insulin sensitivity with thiazolidinones improves psoriasis. So it all seems to be, this state of diabetes inflammation that enhances the psoriasis. What's the evidence that psoriasis actually enhances type 2 diabetes? And again, going back to the Mendelian randomization, can you see the cause-effect relationship between the two?
We did do a MR study. It was a little bit unclear, but we saw some potential causal association in both directions. I believe the only one that was significant was diabetes to psoriasis. I'd have to check that, but I think it produced some interesting results, but we need to have larger power from, I think, we have a new psoriasis GWAS coming out soon, which will hopefully help us to look into that and get more power for the NR analysis.
So, my question's about... Okay, my question is about the COVID Inflammatory Skin Disease Association. So I'm wondering if you control for medication exposure for inflammatory skin diseases. And the reason I ask that is in IBD, for example, we're seeing that steroids that people are put on seem to drive the association for COVID-19. But some of the biologics are supposed to actually protect against COVID-19. And so I'm wondering, because you have a very similar sort of trend in your data, if you're going to look at that or might think about it.
Yeah, good question. So I don't think we looked at steroids particularly, but we looked at biologics because there's a very fact biologics could affect the risk of COVID, and people were wondering, should we stop treatment with biologics during that time? But in our analysis, we looked particularly at IL-17 biologics, which are used to treat psoriasis. We didn't see any association in our results. It didn't affect the correlation between psoriasis and COVID.
Okay, last question.
So if indeed there are some shared loci between psoriasis and type 2 diabetes, did you notice whether the age of onset of those two conditions in your 120 or so patients were similar age of onset?
Thank you for your question. I don't know if you can check that. I should look into that. Yeah, I can see what would be interesting. I should do that.